Hi everybody. This week we study chemical genetics. Again, this week I chose some references from Wikipedia, but I realized now we are getting into more specific topics and it is getting more and more difficult to find general references either from Wikipedia or YouTube. So this week I used more amount of the professional the references like journals or papers. And in chemical genetics part, we are going to study and the difference between genetics and chemical genetics. And genetics approach has a two approach, forward and reverse. So first of all, we are going to study forward chemical genetics and its unique advantages. And then important part of forward chemical genetics is phenotype screening. So we are going to think about how are you going to decide the best screening for your work. And then we move to reverse chemical genetics and its benefits. And then I think it's a time you guys need to think like the problem for your proposal. The problem you need to think what kind of a proposal you wanted to make. Okay, let's move to the forward chemical genetics. So to begin with, let's check the genetic part of biology. Our genetic information is stored in DNAs, and one unit of those information is called as a gene, and the whole cluster of this gene we call genome. And some part of this information is transcribed into RNA, especially this RNA we call messenger RNA, mRNA. And utilizing this RNA, we make a protein. So a very fundamental definition of the gene is one information to make one protein. That is our definition of a gene, right? Once all the proteins are perfectly working, and then we can see the whole shape of our body. So now here, the information is here in DNA, and the end result is shown to us as a phenotype. So we call this as a phenotype, and then this genetic sequence as a genotype. Okay, phenotype and genotype. Then what can be genetics? Genetics is the study of gene, simply speaking. That means genetics is the study of a gene's function. In another word, gene make a protein, then genetics can be the study of a protein's function. We can say that, right? Then how can you connect this gene's sequence, in other words, the genotype, to phenotype? There are two approaches. First approach is, let's say we have a phenotype here, and then somehow we disturb this phenotype. The conventional method was to give very high energy to the life forms. And in this genetic study, the popular model was this kind of a small fly, so-called fruit fly. Professional world, it says the Drosophila. And then we give X-ray or UV light to the fruit fly, and then induce some kind of changes by giving damages. And then check whether that damage is transcribed to the offsprings. If it does, let's say the eye color was changed, and then the same color is transferred to the offsprings. That means somehow their genetic information is changed. Then we have a chance by using this phenotypic change and to figure out which gene is changed. To find out this change is just a sequence change, right? So that sequence change is we call mutation. To find out that mutation, we need to search all the genomic change and then find out which gene's change is directly related to this phenotypic change. So this approach, we started from phenotypic change, and then later we found out which gene is changed. So this approach is called, historically started first in genetics. That's why this approach is called as a forward approaches. Starting from phenotype, figuring out which gene is changed. That's a forward approach. Now, in modern the molecular biology, so we have a lot of new tools how to modify these genetic structures. In other words, this can be called as genetic engineering. So now it's possible, even after we are done with the genome sequencing, now we know all the sequences of species. Then we can choose specific gene by just reading their sequences. And then we have option to, let's say, to delete that gene. If you delete, it cannot make RNA, and eventually it cannot make a protein. Then we check what's happening in the phenotype. So then if you can find some changes, oops, one leg is gone, then maybe you can connect this gene's function is somehow related to leg development. Maybe you can call this gene as leg gene, something like that. This approach, comparing to the previous approaches, for the approach case, we started from phenotypic screening and then later figured out which gene is changed. That's why this is the opposite approach, starting from genotypic change here first and then figure out what kind of a phenotypic change happened. So this approach is called as reverse approach. There is some problem here. We said the total genomic number, the gene number, was much smaller than we expected. That means most likely one gene's function may not be only one function. It may have multiple functions, and then sometimes it has a very unique function during those fetus development. And then after it is born, it may have different functions. If you delete this gene from the beginning, then it cannot make that critical protein. Then the fetus does not have a chance to survive and it cannot make a birth. So that means if this protein has a different function later stage after it is born, then we lose the chance to observe this gene's function. So that means this kind of a permanent gene deleting has significant problem and a lot of limitations for time-dependent study. That's why other method may be necessary. So here, this second approach of reverse approach is, without touching DNA, we just attack only the mRNAs. If you add RNAi, it does not attack DNA, only it attacks messenger RNAs. Then RNAi block the function of a messenger RNA or destroy the messenger RNA, then protein cannot be made, and then we observe what's happening in the phenotype. So in this case, at least the good news is we can select the time point when we wanted to add RNAi, and then just see those effects only for that specific time point. So this is a much more time-controlled experiment. Now let's compare. Here, if we remove the gene from the whole genome, then that's we call knockout. Knock is door knocking, knockout. And this is knockout, but this mRNA temporary suppression is different from the knockout. So this mRNA, siRNA or RNAi experiment we call knockdown. So knockout is removing the gene. Then in other words, if you introduce new gene into genome, then what would you like to call? Knockout? Knock in, yes. So in biology, sometimes once we have one term, then to make it simple and to compare, so we make some related word. Removing gene from the DNA, knockout. Introducing new gene, we call knock in. And then suppress that mRNA function, we call knockdown. To summarize, let's say one is you select your target first and then see what's happening in system. The other is you disturb the whole system and then figure out which is the target, right? So then, which one is which? Target to system and system to target, which one is a fold approach? Yeah, this should be fold approach. You started from phenotypic change and then figure out it's a target. Reverse approach, you select your target first and then somehow change, knock out, knock in or knock down and then check the effect in systems. So this approach maybe you can call this is reverse genetic approach. Now let's compare with the chemical genetics with the traditional or the genetics approach. In genetics approach, especially in fold genetics approach, we introduced a very strong shock to the liposomes. So those shock like UV or X-ray, it damages our DNA. and it changed the sequence of DNA, and then eventually that mutation affected those phenotype, and then that mutation is transferred to the offsprings. In chemical genetics, we use a different shock. We do not use that kind of physical shock to change DNA. We give the shock with a small molecule, so-called drug-like molecule. Then those drug-like molecule, it can go inside the body, and then somehow it change something, its phenotype. Maybe let's say in this example, sexual changes. Some kind of effect happens. Now in this case, now we have a different type of solutions to figure out its mechanism. In genetics case, at least, the gene is changed. The gene mapping to figure out which genes the sequence is changed is a lot of work. But still, that signal is left over in the body, and then it is transferred to the offsprings. We have a chance to the whole genomic sequence and figure out that changes. But problem of chemical genetics or difficulties, if we add this compound inside the body, most likely this compound may interfere or may bind to some protein, receptor or enzyme, but that effect is not transferred to the next generations. It just affects that enzyme itself, and we need to figure out its a binding partner of this small molecule. This process we call target identification. The target is, of course, it's a target of our small molecules. So that this part of target identification and also its validation is quite challenging at the part we need to figure out. So that's why this part we are going to handle in the next class. But still we have a lot of advantages if we use this chemical genetic approach. So let's check the advantages of chemical genetics. First of all, the effect is quite fast. So if you add the compound into biological system, then we can see the effect within several minutes or the hours. If you use siRNA, what do you expect? If you add siRNA, siRNA's function is to block the synthesis of a new protein. Even though we target the same gene and same protein, chemical genetics approach, you can kill those function of the protein right away. But siRNA effect, it's just a block new synthesis. That means already existing proteins still functional until it is naturally degraded. So it may take maybe hours or days until all the proteins are removed. That means siRNA is much more temporarily a controlled experiment. Still, effect-wise, that's slower. Also, chemical genetics approach, you can control the amount to put. So if you figure out the IC50, inhibition concentration 50, then you can control approximately. You want to kill most of those enzymes or 10% of enzyme kind of things. siRNA is a little difficult to control that way. Usually we give enough amount to fully knock down those genes and then wait some time. So in that sense, this chemical genetics is more controllable. And also, this chemical genetics approach is conditional. We can add the compound, and later we can wash it out. And sometimes we can turn on, turn off. That control is much more straightforward than other approaches. Another challenging part of the studying of the gene's function or protein's function is potentially one protein may have multiple functions, and it may have multiple binding sites. And if you totally delete that protein by knockdown or knockout, then we see the whole combined effect. But when we use chemical tools, it is possible, maybe, it can selectively attack only one function or one binding site, and control for one specific other function is potentially possible. This is another advantage of chemical genetics. Of course, these small molecules has a good chance to be developed into drug molecules later. So chemical genetics, the method, is directly related to drug development, the impact. And using this chance, there is a possibility that if you see very novel phenotypic change, then we have a good chance to figure out the new function or previously unknown function of those protein after we get the ID of those target. So these are kind of unique advantages of chemical genetics. Now this is kind of a summary of chemical genetics, especially forward chemical genetics approach. So first step is somehow we need to prepare chemical tools. It can be natural product, or it can be combinatorial library, or it can be marketed drug compounds we can collect from the commercial sources. So different method we can use to prepare these toolboxes. Next step is you need to set up important and interesting phenotypic screening. Once this is set up, then screening should be done very quickly. This quick screening we call high throughput screening. We are going to talk about it later again. After that, the next step is figuring out those target protein, so-called target identification. So this target identification usually we rely on affinity matrix and then protein analysis. This part we are going to handle next week. So using this big picture, now let's think of a step-by-step of a procedure of forward chemical genetics. So first step is you need to define your problem. So this is exactly same as the problem I cast to you for your proposal. You need to think what can be important problem you want to solve. Somehow biologically related or medically related problem, and then utilizing chemical as a tools, and you solve those problems. Once you define the problem, the second step is you need to define or design your screening. Let's say if it's a cancer, then maybe you can start with the cancer cells, and then cancer curing is your screening format. So this is the step of your phenotypic screening, the design steps. After that, you need to think how to prepare your chemical tools. Either you synthesize, or you collect from the commercial sources, or collaborate with a natural product chemist and collect those tools. Kind of a plan you need to make. After that, you actually do the screening, if possible, efficient way, fast way, and cheap way. After your screening is successfully done and you found interesting compound which can induce some expected phenotypic change for your work, and then the next step is you need to identify your target to explain the mechanism of the compound. Once you believe you found your target protein, then you need to validate to confirm whether that target is really your expected target protein. After that, optionally, you can think developing your hit. Here, the hit is the candidate molecule you found from your tools. And utilizing this, the hit molecule, whether you can think to develop this useful academic tool or drug development. So this is general procedure of forward chemical genetics. Among these problems, we are going to talk about the target identification and validation next week, and also how to prepare the chemical tools the next week. So today, we are going to focus on phenotypic screening. So here are some examples of very common phenotypic screening as a model. The first model is yeast. Yeast is a monocellular organism, so it looks like a bacteria, but comparing to bacteria, yeast cells has a nucleus. So that means prokaryotic bacteria, they don't have a separate nuclear structure, but yeast cells has a separate nucleus. That means yeast is much more advanced the structure they have, so it's much closer to human beings. And yeast cell has two different kind of the shape. One is the budding yeast, and the other one is the fission yeast. Budding yeast is a little more the primitive the life form, and when they divide, they make new cells like this. So this is a mother cell, and this is a daughter cell. So this is like a budding structure. That's why we call this as a budding yeast. When this daughter cell becoming bigger and bigger, so it reaches the enough size to divide, then they divide here. Fission yeast, by the way, they make almost looking identical to cells and then divide in the middle and then make two cells. So this division method is much more similar to human beings. So that's why we can assume that fish and yeast is a more developed, advanced structure they have. So some people say, even though those two other species, they have the same name of a yeast, but difference between these two species is much bigger than Pombi to human. Interesting, isn't it? So by the way, this budding yeast we call S. cerevisiae, and this fish yeast we call S. pombi. First of all, these cells are quite familiar to human being. These yeast have been used to make wines and beers and also baking the bread. That means relatively these species are quite safe to handle and very easy to grow, and a lot of their genetic informations are conserved with the human. So that means if we have some protein, most likely those yeast cell has also similar functional enzymes they have. So this is the case. It's one of those earliest the genome sequenced, and then it has about 6,000 genes. And very interestingly, human has about more than 20,000 genes, right? Only 1/3 of the numbers of genes. And many of those genes, if yeast has, we have one copy or 2-3 different kinds of the variations we can assume. Now, another interesting observation is the yeast is a monocellular organism, but they have a sex. We don't call them as male or female, but we call them as A form or alpha form. And interestingly, the sexual forms, they are haploid. That means they have only one set of DNA. It's like a sperm or egg cell status. And when they mate together, and they become diploid. And also interestingly, when they mate, alpha form, they mate only with the A form. So that means A form does not mate with the A form, alpha form does not mate with the alpha form. So we can say yeast cells are not homosexual, they are heterosexual organisms. Interesting, isn't it? When they mated, then they form diploid. In our standard, most of our the cells in our body, they are diploid cells, and only sperm cells and the egg cells are haploid. But in yeast case, most of the cell status, they live in haploid status. Only when they had some environmental difficulties to survive better, they find its partner, and then they fused together, and then they survive their conditions. And another aspect, the interesting aspect is yeast cells, similar to bacterial cells, in addition to the cell membrane, outside of a cell membrane, they have a cell wall. This is a typical shape of the yeast growing. Now, in this kind of a plate, we spread out those yeast cells, and then they grow out. And this white part is high density or high population of those yeast cells. Now, if you spot those compounds, test the compounds here, then you can see the black spots. That is kind of a clear area. The cells are not clearly growing like this. So this is a typical shape of screening the system of yeast, whether one compound can kill the yeast or not. Another very popular the chemical genetic model is a plant, especially this species here, so-called Arabidopsis. This model has been used, the quite popular model for chemical genetics study. First of all, the size is quite small and easy to grow, and also, importantly, all of the known growth regulators, like plant hormones, they are interestingly small molecules, not peptides, not the proteins. Many of those hormones of a plant, they are small molecules. That means it's very easy to incorporate those small molecules into these systems. Also, plant, they collect those small molecules from their root. So it's very easy. If you add a small molecule here, then they can be easily absorbed to the body. That's another big advantages. Genome sequence is fully sequenced quite early time. It's already 15, 16 years ago. And it's very easy to study those genomic scale of the functional study of this species. And even though they are quite small and they don't have a brain or they don't have eyes, but gene number wise, it's almost a comparable size to humans. So we should consider plants are quite advanced form of lives. Another interesting the model for genetics and chemical genetics is C. elegans. C. elegans is only several millimeter long, the small, the worm, and it is a really elegant system. It has a total about 1,000 cells, but starting from the one egg cell, when they divide 1 into 2 and 3, 4, and all those division and making those development into full size, every cell has a defined role. Even you can give a number of those 1,000 cells with the different numbers. So those developed paths is very well conserved, and it's an excellent model to study one compound's effect or one factor's effect. How does it change to which part of the body is very clearly defined. That's why this model is so elegant. So elegant, that term is reflected to its name. So that's why we call it C. elegans. Also, the lifespan is quite short. So it's only 2 weeks of a lifespan. So this is one of those best models to study those lifespan of one species. It's quite small and it's relatively transparent. We can look through inside, and even though they don't have the eyes and nose, they have quite high number of genes, almost a comparable number of genes to human. Human has a little more than 20,000, we believe. Now the C. elegans has almost 20,000 genes. Genomic sequences has been done, so it's very easy to control. And another important thing is RNAi was discovered in this other life form in the beginning. So that means RNAi screening is very easy to apply to these other systems, C. elegans. Another very popular model is a Drosophila. So especially this Drosophila, so-called fruit fly, has been used in genetics almost 100 years of time so far. And they have, of course, sex, female and male, and they have a relatively short life cycle. And gene number is a little smaller than the C. elegans, but quite high number of genes that they have. And gene sequence is already done. And another good thing is we found that RNAi is also working very well without special treatment to this insect cells. So traditionally, we had accumulated the data from the conventional or classical genetics study about those genes, and then now we have a chance to compare our chemical genetic data with this genetic data together. The examples we have seen so far is, in a sense, they are a little lower, less complex system than human body. So they were monocellular system or a little different shape of a plant system and worm system. So it's not vertebrate. So in a sense, maybe they are mimicking to some extent our the functional genomic the role, but somehow it's different from ours. So maybe best way, most direct way is utilizing our own cells. How about the human cells? Then human cell has exactly same genetic information inside. Then we can say they are part of us. That may be most straightforward approach, right? So then using those human cells, then we can do either toxicity test or morphological change test. For example, if the cells are forming this kind of tubes, this is a muscle tubes, then this muscle tube, they have a multicellular structure inside here. And by giving some chemical the tool or drug compounds here, if you can induce this cleavage of those tubes into individual cells, it's a dramatical morphological change. In other terms, it's a shape changes. Then it's a very easy and interesting observation or phenotype screening. So utilizing this system, one way is you can make from this differentiated cell into more primitive cells, so-called maybe stem cell-like cells. So this kind of the possibility is there depending on how you define, how you set up your screening system. While this is a quite interesting observation, but in a sense, this is just a cellular experiment, then one of those the critical questions should be how to connect this into the whole body effect later. So that's one problem. The other potential problem is whether your chemical tools, whether they can get inside of those cells and then have a chance to attack the target protein. This problem we handled before in terms of the molecule's drug likeness. You remember? The Lipinski rule is talking about this kind of property, whether the compound can get inside the body or not. This is the question. If you are really worrying about those cell penetration issues, one potential possibility to remove that problem is, why don't you just remove whole membrane structure? You just take the whole cells and make an extract. Then this system does not have any membrane structure. This system we call cell-free system. So that means cell structure is partially broken. All the components are mixed as one pool. So that's why you don't need to worry about the accessibility of your compound crossing those cell membrane. So in this interesting example, you make a cell extract first. So to make sure you have enough proteins in there, usually you use egg cells. Any egg cells, they reserve a lot of energies and protein other cells inside there to support their next generations. So that's why usually their protein content is much higher than other cells. And this example is utilizing frog egg cells. So take those frog eggs, make a cell extract, and then add red dye. In this case, this red dye stains microtubule. So they form proper microtubule structure, and then you can add extra DNA the structures. What can be the highest contents DNA among different cell types, do you think? Question again. If you choose one cell which may contains very high portion of DNA, what cell can you think? Egg cell? No, egg cell has a small nucleus and a lot of proteins. Yes, it's sperm cells. You can assume that the head of those sperms, it has a tails, but head, maybe almost those head is filled with DNAs. Interesting, right? So that's why the source of those the DNA usually we take from fish sperms. Collect a lot of sperms from fish, then maybe 80%, 90% of those contents are all DNAs. So collect those DNAs and add those DNA into this system. Then they mimic cell division, and then DNAs are condensed in the middle here, and the microtubule is attached to this system. So this kind of proper cell division is quite important for cell growing and cell proliferation. So this is your screening system. Now, if your goal is you want to disturb this cell proliferation, cell division, then maybe you can stop cancer cells very fast growing. Then by adding different compounds, you can see different effect. If the compound is indirectly attack those system, then microtubule is not properly oriented like this. They are hanging around like this. And DNA is not properly condensed. They are kind of partially spread out. If the compound effect is attacking microtubule, then microtubule is shortened, and then the DNA is still condensed in the middle, but microtubule structure is interfered. If you have a very strong microtubule acting compound, then maybe DNA is not affected, but whole microtubule structure totally gone. So this kind of dramatic changes during the cell division can be monitored using these so-called cell-free systems. Quite interesting system, isn't it? Next system I wanted to introduce to you is zebrafish. This is one kind of fish, and if you think about their structure, fish is a vertebrate. And if you grow those fish from eggs, this is one egg state, and then divide those cells into 2, 4, 8, and then further and further, and then after about 1 day of time, it has eyes and brains and it's a vertebrate here. So it is 1.5 days. Already it has quite a good shape of those embryo, which is very similar to our own embryo development shapes. Now, one of those very popular model is a zebrafish, and as I said, this is a vertebrate. Very similar structure they have. They have a brain, heart, and bone, kind of different, the complicated structure, very similar to the human beings. And they are quite small in the embryo status, so they can get in very comfortably in 96 well, very easily. And they can be very prolific. So hundreds of those the embryos can be produced by one pair of those parents, and they are completely transparent. So even without any physical dissection or killing them, we can monitor what's happening inside their body. It has been quite difficult to study to some extent because their gene sequence has not been done until recently, but finally it's done a couple of years ago. Now those zebrafish whole genome is fully sequenced. So far, RNAi is not working very well, but there is another technique, morpholino, kind of antisense technique has been used. Now, other technique is introduced into this system too. Gene number is comparable or maybe even a little bigger than human. So zebrafish is quite advanced model system, we can say. Lifespan, the early embryo development is only within several days. It has forming up quite important organs very quickly. So it's very easy to study for those early stage of those genes functions. But lifespan of this fish itself is quite long. It's about 2 years. Then that's almost similar to mouse, another very popular animal, the model in the lab. Then maybe this model is a little difficult to use for lifespan kind of studies. If your PhD is only 4 years, then monitoring this lifespan, whether they are live longer and shorter than average, then it takes multi-years of the experiment. So you should think carefully which model you should choose, kind of things. At least for this zebrafish model, I found a very interesting educational the movie. So follow this, you will see those professional the demonstration of introduction of a zebrafish as model system for drug discovery.
Drug discovery scientists are increasingly excited about a new research model called zebrafish, which have nothing to do with zebras. The direction of the stripes is different. Zebrafish are tiny, inconspicuous fish that live in big numbers in sweet and brackish waters on the Indian subcontinent. Aquarists love them because they are undemanding and thrive in almost all conditions. For drug discovery, zebrafish themselves are not that interesting, but their offspring are. A zebrafish pair generates about 200 eggs per week, which hatch within 2 to 3 days to give zebrafish larvae. Technically, these are not considered laboratory animals. They do not move and do not feed, and they are so tiny they can fit comfortably in 96 well plates, the typical assay format for bioactive compounds. If you wonder about the relevance of testing drug candidates on a fish larva, you are not alone. Zebrafish of any age do not look very much like humans. 70% of human coding genes have an equivalent in zebrafish. Not too exciting if you consider that this number for mice is around 85%, and for chimpanzees, greater than 90%. And they are not a perfect match either. To understand the interest of scientists, you have to remember that the only models with similar scope available so far were a fruit fly and a worm. So a fish, being a vertebrate, is a gigantic step forward. One important aspect is that embryonal development is very similar throughout the vertebrate kingdom. Zebrafish embryos grow their organs the same way as humans, only 20 times faster. Instead of testing potential drugs for efficacy or safety on cultured cells, as is common today, zebrafish offer those cells within a working organ, and that organ can interact and communicate with all other organs around it, creating a multi-organ culture model with a similar body plan and functional structure as a human being. The effect of a compound can be tested on a beating heart, a metabolizing liver, or a secreting kidney. Moreover, in the last 2 years, many methods have been published that simplify the genetic engineering of zebrafish. You can simulate human diseases by gene mutations, Or human genes can be put into zebrafish to see how potential medicines work on those proteins. Targets of interest can be genetically tagged with fluorescent proteins to track the effects of compounds visually through the larva's transparent skin. You can generate large databases containing effects and side effects of known drugs that can be used to classify novel compounds. With the advances in genetic, imaging, and data handling technology, it's not surprising that the field of zebrafish research is exploding, and Roche pRED is actively pursuing many possibilities for both efficacy and safety testing in a variety of models and setups. Meanwhile, even aquarists have taken an interest in the exciting implications of zebrafish engineering. Glowfish is a zebrafish that has been genetically modified to express fluorescent proteins in all tissues. It comes in a variety of bright colors and glows in the dark. The possibilities are endless, but we prefer to stick to drug discovery for the moment.
How was the movie? It's quite good, right? And this is the source of those movies. Now, this is first part of today's lecture. This is for the chemical genetics, and also I showed you some examples of potential phenotypic screening. I think it's a time you need to seriously think about your proposal topics, and using these examples, this is one big part of your design potentially can be. So what screening system would you like to use or to think about it? In addition to the examples I showed you, you can think any new phenotype screening you can propose. The key issue is whether this phenotypic the screening setup is easy enough to use and easy enough to monitor the changes. If it is very subtle changes, it's very difficult to follow. And if it is very difficult to model, let's say I want to study lions and I wanted to have thousands of lions in my the laboratory work. It's too messy and too expensive way, right? Then you can think, what can be manageable size? Assuming that you want to test hundreds or thousands of or 10,000s of molecules you want to test in this phenotype screening, what kind of a system would you like to adopt or to invent or to propose? Okay, think about it. So this is end of those first part of today's lecture.